Hello, grade 12. So today we are looking into DNA and RNA. Okay. So what is important on DNA and RNA is to understand the structure, the location, and the function. Okay. So let's start here with the discovery of DNA. We need to remember that the DNA was discovered, the 3D DNA double helix was discovered by Watson and Crick. Okay, so these are the guys that you must remember, the one that discovered the structure of DNA. Okay, then let's move on to the location. DNA is located in the nucleus. So the nucleus of the cell, you find DNA, and also in the mitochondria. And on the nucleus, we call it nuclear DNA, and the mitochondria, we call it mitochondrial DNA. But these two here, you can find them on both plant and animal cell. Okay, so you find the DNA in the nucleus and mitochondria in both plant and animal cell, but for the chloroplast, you only find it in the plant cell. Okay, because we don't have a chloroplast in animal cell. But what are the functions of DNA? DNA controls the functioning of the cell. Okay, that's number one. It also regulates the functioning of genes. But we need also to understand what is a gene. Okay, what is a gene? Okay, so a gene is a segment of DNA, DNA that code for synthesis of specific protein. Okay, so that's what you must remember. But you find that when you move on to genetics, we'll refer to this as segment of DNA that controls specific characteristics or traits. Okay, so that's what the functions are. That's what a gene is. Okay, so you must always remember. So when you're talking of a control specific characteristics like height. Okay, so we are talking about this as types of genes that you can find in a cell. Then also another thing of functions of DNA is it passes on hereditary characteristics. That's why you see you look similar to your parents and also grandparents. It's because of the DNA and the characteristics that are carried on from one generation to another generation. Okay, now let's move on to the next slide, where we are looking at the structure of DNA. So the first thing that you can see here, the DNA is coiled, but also you can see that they are double stranded. So what is it that we need to state there? DNA is double helix. So we need to understand that the structure of the DNA is double helix. Double helix meaning to say it's coiled. Okay, as you can see there. Then also, as you can see on the diagram 2 here, you can see that we are seeing two strands of DNA. So we are saying DNA is double stranded. Okay, double stranded. That's what you see. But as you can see, we can see that there's a long chain. And this long chain that we see here, we refer to it as a polymer. Okay, we refer to it as a polymer. And we are saying this what you can see something here. We call these are the building blocks. And this building block, we refer to it as a nucleotide. Okay, a nucleotide. So building blocks of your DNA is a nucleotide, and we refer to this as a monomers of the DNA structure, which is a nucleotide. But a nucleotide is not only it's made up of three things here. We have got the phosphate group, that's number one. Okay, so phosphate group, as you can see. Then number two, we see that we have deoxyribose sugar, that's number two. As you can see that these are the sugars, the deoxyribose sugar. And lastly, we have got what we call the nitrogenous what called nitrogenous base connected to a nucleotide. So a nucleotide consists of phosphate group, deoxyribose sugar, and nitrogenous base. But on the nitrogenous basis, we can see that we have got ATGC. We have what called adenine, okay? Adenine, and adenine, that's our A there. Then we have got number 2, we have got what called thymine. Okay, that's we are referring to as T. Then number 3, we have what called guanine. Guanine, okay. Guanine, which we are referring to as G there in the structure. Then lastly is cytosine, which is actually referred as C on the structure. But as you can see, the strands are bonded together. They are linked together. The two strands are linked together to form a double strand. But what are they linked together with? They are linked together with what called a hydrogen bond, weak hydrogen bond. Okay, it's called a weak hydrogen bond that links what you call these two strands. But they are linked through complementary base pairing. We call it complementary base pairing, complementary or specific, the specific base pairing. What do we mean by that? We are saying specific base pairing. What am I writing now? Specific base pairing. So specific base pairing, we are saying on A can only pairs with T, and C can only pair with G. Okay, so adenine can only pair with thymine, and C can only pair with guanine. Okay, that's what you must remember on the structure of DNA. Now let's move on to another nucleotide that we must remember, which is the RNA. What is RNA? It's ribonucleic acid. Ribonucleic acid, and RNA consists of nucleotides. And we know now the structure of the nucleotide that we have got. We have the phosphate group. We have the sugar. We also have what called the base. But on this sugar here, it's unique now because this sugar now is no longer deoxyribose sugar, it's ribose sugar. That's what we see. But the phosphate group and the nitrogen bases remains what called the same. But there's something also special about RNA, which is one unique base that you will find, and this base here we call it uracil. So these you find them on both RNA and DNA, but uracil replaces thymine. So replaces thymine. So it replaces thymine in DNA. So in RNA, you find uracil. In DNA, you find thymine. Okay. Now let's move on to the next to understand the types of RNA. It's unique now because we have got 3 types of RNA here. So the types of RNA. And these types of RNA, the first one is the messenger RNA. And this is the one that you see during DNA transcription process of what called protein synthesis. Okay, so messenger RNA carries the code, which is the message, okay, the code for protein synthesis from DNA and ribosome. We'll explain this, why we have RNA carrying message to the ribosome. We'll talk about this when we're doing protein synthesis. But this is what you must remember. Messenger RNA, it carries the code from protein synthesis from DNA to ribosomes. Then the ribosomal RNA, it is the site for protein synthesis, because we know that the protein synthesis occurs on the ribosomes. So ribosomal RNA is actually the site for protein synthesis. Then transfer RNA, it actually brings amino acids to ribosomes to form protein. So here, this RNA here, the main functions of actually your RNA it plays a very crucial role in protein synthesis. Okay, so in protein synthesis, that's why your RNA is busy there. So the location of the RNA, where is it located? You see that it's located in the nucleus. What is located in the nucleus? Messenger RNA, because messenger RNA is formed in the nucleus. So we can say the first location is nucleus, but it then takes the message to the ribosomes. And where are the ribosomes? The ribosomes are in the cytoplasm. So it goes to the cytoplasm. So you will find this also in the cytoplasm. What about ribosomal RNA? Where do you find it? You find it in the ribosomes, in the cytoplasm. So you can see that we have got ribosomes now, and what is it? Also the cytoplasm, and transfer RNA is lying free there, carrying amino acids to the ribosomes. So it is located in the cytoplasm. So you can see that the main location that we are talking about here is the nucleus, is the cytoplasm, and it's also the ribosome. This is where you find your RNA. Okay. Now, let's move on to the structure of RNA. As you can see there, is different from the DNA, where you see two strands connected together. But in this case here, we are saying RNA is single-stranded. Single-stranded. So it's single-stranded, and remember what we said, it's also made up of nucleotides, the same nucleotides, but the difference here is on the sugar, and also one of the bases, which is the uracil. In this case here, I didn't put it. Can I just maybe replace this with a U? Okay, let me just replace this with a U. Okay, so that's our uracil there. Okay, because we don't find, let me also replace this with a C, so that we can have the four what called nitrogen base that we will find in the RNA. So it's adenine, it's guanine, it's uracil, then it's cytosine. Okay, these are the four nitrogen bases that you'll find in the RNA. So it's single-stranded, and as you can see, it's made up of it's a polymer. Again, it's a long chain, so it's a polymer. The long chain. It's made up of monomers of nucleotides. Monomers of the nucleotide. Okay, so you must always remember that the monomers of your RNA and DNA is a nucleotide. Okay, and the one nucleotide consists of one phosphate group, and one nitrogenous base, and the sugar. So it's a sugar, which is the ribose. Then it's a phosphate group. Then is a nitrogenous base. Okay, this is the nitrogenous base. Okay, now let's move on to the second slide, where we are looking at the comparisons, where we compare the two, DNA and RNA. So you find that there are some similarities, because they both contains sugar alternating with the phosphate. So they both contain what called the phosphate, and also contain nitrogenous bases adenine, guanine, and cytosine. Okay, so these are the common bases that you find on both. They also both play a role in protein synthesis, because remember, the code for protein synthesis is actually obtained from the DNA. But the DNA cannot leave the nucleus, so it has to pass that message or that code to messenger RNA to take it to the ribosomes, which is the site for protein synthesis. Now, let's look at the differences, as you can see. But from the difference, you can see that on the left there, we see a double-stranded. We see a double helix. So here we have got the double helix. We have double-stranded DNA. But on the right there, we see a single-stranded DNA. It's a single-stranded RNA. So single-stranded. So this is RNA. This is your DNA. Another thing that you see, you can see that you are spotting what called thymine there. And as I've indicated there, let me just put the remaining what called bases there. It's uracil and cytosine. Okay, so what you will see there, you will see presence of thymine. So thymine is found on the DNA, but on the RNA, you find uracil. Okay, then the sugar, as you can see from the sugar there, we have got deoxyribose sugar. So deoxyribose sugar here, let me circulate it. But in this case here, on the RNA, we have what called ribose sugar. So we have a ribose sugar. That's another another difference that you will see there. And yeah, so this is what you must remember when you're looking at the differences and also similarities of DNA. Now, let's have something to just to see if we understand this chapter. Now, we have an activity there where we ask you label 1, 2, 3. This is what is happening with life science. So life science, you need actually to be observant, because as you can see here, we need to label 1. How are we going to know which nitrogenous basis is? Because we can tell that this is a nitrogenous base, this is a nitrogenous base, but how do we know the name of it? So as you can see here, we can see that there's a G here. So it means that we're going to have a C here. But you can see their structure. And as you can see here, this is a thymine. And it looks the same as this, what called this base here. So it means that this is going to have thymine. But we know that there is complementary base pairing. So our T1 is going to be our adenine. This is what we see. So we are comparing with the base that are actually labeled here, and we can now come back and answer the question. But what about the 2? 2 is a sugar, but remember, we are looking at DNA here. So it's a DNA structure. That's what we're looking at here, because it's double-stranded. So what is 2? 2 is a deoxyribose sugar. Then give the number of nucleotides shown in the diagram. So number of nucleotides, as you can see here, we can that this is a nucleotide. So this is a nucleotide. A nucleotide, remember, consists of consists of phosphate group, sugar, and nitrogen base. So you must know that one nucleotide contains one nitrogenous base. So we can simply count nitrogenous bases here. So we can say 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. But also we can only count also one side of the strand, where we can say 1, 2, 3, 4, 5. Then you just say 5 times 2, it will give us 10, because we know that they are actually in pairs. Okay, so this is 10 nucleotides. Name 2 places in an animal cell where this nucleic acid may be found. They said what? Animal cell. So what is guiding you here is animal cell. So in animal cell, we know that we can find this in the nucleus, then also in the mitochondria. Then let's move on to what is the natural shape of this molecule. Natural shape, we know that is double helix. Okay, then draw a nucleotide with the nitrogen base adenine. They specified adenine. So you can now simply draw your sugar there. Then we have adenine there. So that's indicate there with the red. So that's a nucleotide. But remember, you must label the phosphate group, you must also label the sugar, and you must label that that's adenine. Okay, so this is the end of the class. I hope you have enjoyed, and actually I wish you the best in your studies. Thank you so much. Until next time.